Starting right now, listen for that. Bite you by Gary's Brainland. Okay, so what are we doing here? This is the lab where we're going to measure if photosynthesis and cell respiration is happening. So let's walk through a couple things. So the pre-lab has this liquid that is called a carbon dioxide indicator. Other labs do it with something called bromophenol blue. All right? So here's the liquid, and let me just on camera get a little witness. Like, what is it? I'm walking around to different groups. What colors look like?
Red.
Now, your meter, you guys, actually, you know what? Here's the meter. We'll put the meter right here. And you guys, one of the things you're doing with the lab is reading, using that meter to tell. So let me put this so you guys can see. All right? So, you're our witness. What is it? What number do you have?
It looks like maybe 8.3.
About 8.3? Okay.
It looks like maybe 8.3. It's a little lighter than that.
Okay. Okay. Second verification. We're out of camera view, but I put it on someone's table to tell us. Come on, quickly, go. 8.5. Hey, everyone can be writing this number down. We'll get another verification. What do you got? So, for those who are watching the video, I'm walking around and putting the cups on the student tables to get a quick little reading. What do you got? 8.5. All right, two that say 8.5.
Definitely not that bad, but okay.
Okay, so now.
So where do we write this down?
So, where are we going to write this down? Excuse me. Right here. This is the before. If you want to draw a little cup, that's fine. And now, it's time to. Yep, go ahead. You guys can watch what's happening, and I'll take it up so you can see, does it change color? Just keep going, just keep going. Don't forget to breathe, and don't inhale. Keep going, this is live. Oh, guys, are you seeing a color change?
It looks orange inside the yellow.
Keep going.
All
All right.
Can I stop?
Yes, you can stop now. All right, so I'm going to first put this on so you can see on the camera. All right, we're going to get rid of the carbon dioxide dispenser. Meaning throw the straw away. All right. So I'm taking this to a group now. Where's your They're giving us a reading.
Can sit down?
You can sit down. 7.3. We'll get second verification. 7.3. Are you saying 7.3 because they said 7.3?
No, we were thinking that before.
Third one. So you guys, again, you're going to use. You now have your thing to write this right here. 7.3. Okay. So you'll write that right here. You can answer these questions. So look, so now you can see, and it will show up better on the video here. We've 7.3, 8. What did we just do? What happened to the pH? Whoa, wait a minute. Whoa, what do you mean increased? It went from 8 something to that. Do you remember that? Let's remember the pH scale.
Isn't there like a low usage that's more acid?
Yes, it became more acidic. Okay. Now, how does that fit into this lab? So first, let me kind of give you the makers of this lab. Is from a group called Biorad, right? And we're going to take beads, algae or algal beads. They're in these little. They made these, and we put them in a little tube, like this. So let's see if I can actually show it. So there's 3 in there, all right? And what we're going to do is you're going to get 2 of them, and one of them, you're going to do this. And just And the other one is going to go back there where the light is. So, first mistake students make is, oh, the light reaction, dark reaction. That's not what we mean. Those are the two aspects of photosynthesis. It's the light environment or the dark environment. Then you're going to have to make predictions. So we have to let it sit and take a measurement of using this. So again, you're going to have that. This will be under light. Hey, let's add some light. One's under light, one's not. And then every 8 minutes you're going to record. You're going to record this in your data table. Now, during those 8 minutes, that's when you'd be going back here and keep working on the pre-lab, the notes that help you. That's a good picture. We like that picture. All right? You should be able to answer those. And then we want to start thinking about do the EDD, your title, your hypothesis, and then, of course, the graph. And let me remind you, with the graph, we had to add this little part here, so it's 40. And I will go ahead and tell you, we probably will do a 48th minute if time permits, but we'll still graph based on 40. All right, so let's get into, I want to get into the nitty-gritty of what we're actually doing. So first, you already have the number one's been done for you. You're going to have to do number two. So this is where we got to look back at our lab setup, and then I'm going to close this video here. So here's the deal. You have in here, you've got a trash can. You have the chemical indicator, the carbon dioxide indicator, the aluminum foil, your 2 sets of beads. All right? 2 sets of beads. Now, and you got 2 pipettes. This first one is to remove the liquid. So let's kind of walk through what you have to do. All right, now notice that it's got one tapped in the top, so sometimes you might have to do this. Tap it, shake it, and get it back down. And, all right, so there's the 3. So what you're going to do, and you're going to have to do this twice. You're going to, with the unlabeled one, you're going to basically suck out the liquid and put it in. Keep going, suck out the liquid. You want to get all of it. Now, you want to make sure that you still have the beads in. You don't want to take the beads out. If they pop out, I have a tool that will help you get that in. So, that's really easy. You're going to have to, then, you're going to take the chemical indicator and fill it back up. Close it, and then do it for the other one. All right, let me just kind of, cleanup's going to be really easy because during the first 8 minutes, you're going to go take all this setup back. You're going to wipe this out with a tissue or something, and then we're ready. One is going to go back to where the light goes, okay, and the other one's going to go here, and you're going to take your reading. So what we need to do now, and before I close this video, is get your setup, get your initial reading, and then I will be I'll be doing the clock. You'll see this up on the board, and it will count. And one of the things we also want to do is we get close to like a minute, one of your people go back to the light, and you have this. So this will be at your table, so one's going to do the light, and then I have another one of these back there. So you're going to have to split up your group to get the readings. And you keep recording it. In between, you are doing the questions. All right, and that is our algal bead lab. Now, here's the thing. Why are we using pH? The question to be thinking about, which we're not going to answer in this video, is what happens when carbon dioxide is in water? Read the background, see what happens. Can you tell if photosynthesis is taking place? Is cell respiration? Does algae do cell respiration? Yes, all living things do cell respiration. So those are some of the things to be thinking about when you're answering the questions, going through the background. All right, so now we're going to go ahead and start the lab, and I might do another video of like people in action. All right, brought to you by Curious Maryland. And now it's time to do the lab.